We are going to start our basic science class by discussing the scientific method. The scientific method was developed by Sir Francis Bacon many hundreds of years ago and absolutely revolutionized the practice of science. I will point out that the scientific method starts with observation and curiosity about the observation. Generally, the scientist then, through some level of ingenuity, makes a hypothesis or a guess, prediction, about what he thinks the cause of the observation is. He then performs an experiment and makes more observations, and those observations usually lead to him either accepting or rejecting his hypothesis. If he rejects his hypothesis, he makes another hypothesis and starts all over. If he accepts it, this material moves into what we call a body of knowledge. And then generally it's investigated by outside groups, etc. And if the further evidence does not support the hypothesis, it generally goes back to the drawing board here. But if it does support, then the hypothesis can either become what is known as a theory, in which there's a lot of additional support, or with a lot of additional support and observation, it can become a law. So I want to now go through these steps of the scientific method in more detail in order to help you understand how one goes about performing this process. So here are the steps of the scientific method. The scientific method involves a series of steps that are used to investigate a natural occurrence. We will look closely at these steps and the terminology you'll need to understand before you start a project in science. And you at this level may end up doing a science project for the science fair this year. The scientific method includes developing a question, observation and research, formulating a hypothesis about the observation, the experiment, collecting and analyzing the results of the experiment, drawing a conclusion, and then communicating the results. Now, as you could see from the last slide I showed you, we started with the observation and research. But many times it can start out as a question when one observes something in nature. The first problem is to develop a problem or question. And what that is you look at the world around you and look at a situation that can be solved through experimentation. That is, that can be put into something that you can do in a laboratory or in nature. The second step is to make observations and research about that topic that you're interested in. This way you'll find out about what's already known, so you don't ask questions or do research on things that are already known, but you can use what's already known in order to take it a step further. Once you've done the research, you need to formulate a hypothesis. And a hypothesis is a prediction of a possible answer to the question or problem that you have derived from your observations. For example, if soil temperatures rise, will the plant grow more quickly, or will plant growth increase? So you're wondering about the relationship between the temperature of soil and the growth of plants. The next step is to do an experiment. Doing an experiment means to develop and follow a procedure to answer the question and to look at the hypothesis that you've generated. You have to have a detailed list of materials that you're going to use, and you must be able to measure the outcomes. The outcomes must be quantifiable. Once you perform the experiment, you will collect the data and analyze these data. If the experiment doesn't work the way you want it to, you may have to modify your procedures. Usually you do not do just one experiment, but you'll do a series of 3 to 6 to 8 or 10 experiments to make sure that your results are consistent. When you do these experiments and you collect the data, you want to make sure that you include this data in graphs, tables, and if necessary, take photographs of observations so that they can be shown to other observers. Once you get the data, you look at the data compared to your hypothesis. And then the conclusion is either the fact that you accept your hypothesis or you reject it. By accepting or rejecting, you can also make recommendations in your study for further study and possible improvements in the way that you can take a look at the situation that you've looked at. Lastly, in order for the data to be used, you have to communicate the results. You have to be able to present these results to an audience and be ready to answer questions from an audience about your results. Now let's take a look at an example of the scientific method and see if you understand or help you understand how this scientific method works. Now the problem or question is that one day John was sitting there watching his grandmother, bake bread, and he asked his grandmother what makes the bread rise. And she explained that she puts yeast in the bread, and the yeast releases gas as it feeds on sugar in the bread. Now John's wondering if the amount of sugar used in the recipe will affect the size of the loaf of bread. So if the yeast have more to eat, will the bread get bigger, or does it not have any effect at all? Just a little side note, you have to make sure you understand the difference between the word effect and affect. Effect is usually a noun, and affect is usually a verb. The effect of sugar amounts on the rising of bread. How does the sugar affect the rising of bread? This is actually a situation that I have a great deal of difficulty remembering from time to time, and so I like to make sure people understand the difference between effect and affect. So first John goes out and he researches the areas of bread making and baking and fermentation and tries to come up with a way to test the question. This little guy John here gets an idea every once in a while and raises his hand. He keeps all of this information in his scientific notebook or journal. Whenever you do science in a laboratory, you have to keep a laboratory journal where you write down your research as well as the methods you use and the results you get. Your lab notebook is probably one of the most important pieces of paper or books that you'll carry around with you when you work in a lab. John then talks to his teacher, and she gives him an experimental design diagram to help him set up his investigation. And this is just an example of what an experimental design diagram might look like. First of all, you have a title. It's the effect of, in this case, sugar on the rising of the bread. Now, I'm going to take a second here to point out that sugar in this case is called the independent variable. The independent variable is the variable that the experimenter manipulates. Let me say that again. The independent variable is the variable that the experimenter manipulates. In this case, John is going to manipulate the amount of sugar that's put inside each loaf of bread on the size of the loaf of bread. That's called the dependent variable. The dependent variable is the variable that the experimental observes. He does not control the dependent variable, he observes the dependent variable. The dependent variable is dependent upon the choice the experimenter makes on the independent variable. The hypothesis is if, and this is the planned change in the independent variable, if increasing, you want to know if increasing the amount of sugar in the dough causes or results in a change, an increase in the size of the loaf of bread. So the independent variable is the amount of sugar. And with this, you have to decide, he's going to do 4 levels of sugar. One of those levels will be called the control level, which we'll talk about in a second. And then the other 3 levels are levels that are different than the control. And then he's going to do a number of trials with each. So maybe he'll bake 3 loaves of bread with each level of sugar. Then he's going to say how he's going to measure the dependent variable, which is the size of the bread. And let's say we'll measure size by volume of the bread. There are also constants that have to be taken into account. For example, the temperature at which the bread is baked, the amount of yeast that's put in the bread, the amount of flour that's put in the bread, what kind of flour. All those things have to remain constant. So the only thing that changes is the amount of sugar. So after talking to his teacher and conducting further research, he comes up with a hypothesis. And his hypothesis is, if more sugar is added, then the bread will rise higher. So that if more sugar is added, the size of the loaf of bread will be larger. The hypothesis is an educated guess about the relationship between the independent and dependent variables. The relationship, in this case, between the sugar and the size of the loaf of bread. These variables will be defined in the next few slides. And I went over this once, but we'll go over it again. The independent variable is the manipulated variable. It is the variable that's manipulated by the experimenter. It is the factor that is intentionally varied by the experimenter. So John is going to use these levels of sugar. The dependent variable, or responding variable, is the factor that may change as a result of the changes made in the independent variable. In this case, it's the size of the loaf of bread. His teacher helps him come up with a procedure and a list of materials. She discusses with John how to determine a control group. Well, there's a new word. Let's see what control group is. A control group in a scientific experiment, the control is the group that serves as the standard of comparison. So it's the usual amount of sugar, in this case, used in bread. The control group may be set to something like a no treatment variable in a treatment experiment, or an experimenter selected group. The control group is exposed to everything else is the same as the experimental group, except for the variable D being tested. All experiments should have a control group. Because his grandmother always uses 50 grams of sugar in a recipe, John is going to use that amount of sugar as his control group. John's teacher reminds him to keep all of the factors the same so that any observed changes in the bread can be attributed to the variation in the amount of sugar. The constants in an experiment are all the factors that the experimenter attempts to keep the same. And we talked about these before, things like the amount of yeast, the temperature of the baking, the type and amount of flour, etc. Other ingredients in the recipe, the oven, the rising time, the brand of ingredients, cooking time, type of pan used, air temperature and humidity, where the bread was rising, oven temperature, age of the yeast, They thought of a lot more parts than I did when I was thinking about this. So John writes out this procedure for his experiment along with the materials list in his journal. He has both of these checked by his teacher, where she checks for any safety concerns. And that's important to make sure that you talk to someone who's done these kinds of experiments to make sure there are no safety problems that you might get into. Trials refer to replicate groups that are exposed to the same conditions in an experiment. So the term trials is repeated observations or replicated groups. Replicated just means doing the same thing over and over again the same way. John is going to test each type of sugar 3 times. So he's going to have 3 trials for each level of sugar. John comes up with a table that he can use to record the data. John puts all his materials together and carries out his experiment. And here's his first set of data. And what you want to look at is the average size, which is over on the right, and compare that to the amount of sugar, which is over on the left. The amount of sugar being the independent variable, the average size of the 3 trials being the dependent variable. And you can see that the control group appears to be the largest, but is fairly close to the 100 gram group. He examines his data and notices that his control group was the best, but not really that much better than 100 grams. So he decides, first he rejects his hypothesis because it's clear that more and more sugar is not necessarily resulting in bigger and bigger loaves of bread, but he decides that he wants to find out what's the best amount between 50 and 100 grams. And so he decides to look at various amounts between 50 and 100 grams. So he gathers his materials and carries out his experiments again. And these are the results. You can see now he's gone from 50 to 90. And the size again is on the right here. And you can see that it appears as though 70 is meaningfully better than any other size of bread. Amount of sugar for the size of the bread. So John finds that 70 grams of sugar produces the largest loaves, and he accepts his hypothesis. I didn't see that he had formulated a new hypothesis. The new hypothesis would be something about what he believed between 50 and 100 grams, which he didn't really state. So I'm not sure what they're talking about here when they say accepts his hypothesis. John then tells his grandmother about his findings and prepares to present his project in science class. So that's how we go about the scientific method. In your homework, you'll find, in the handout, you'll find 3 examples of experiments that were performed with various questions. And on the first quiz, you will find that I will take a look at whether or not you understand this stuff that we just talked about. And so it's important for you to make sure you understand these things and bring questions to class. Thank you for your attention.